So muscular contraction is important to every activity that we do. But how does it work? The answer is that we owe it all to a little chemical called adenosine triphosphate, or ATP. ATP is made up of three parts. It's nitrogen base, or the adenine part, a 5-carbon simple sugar called ribose, and a third portion, which is 3 linked phosphates. That's where the tri in triphosphate comes from. When the enzyme ATPase breaks this bond, energy comes out, and then we're left with adenosine diphosphate and an inorganic phosphate. And now what do we do? Before we go any further, chemical reactions are required to break the high-energy chemical bonds between molecules. Enzymes are the catalysts of these reactions, and substrates are the reactive molecules activated by these catalysts. Basically, an enzyme fits into just one specific substrate, like a lock into a key, forming the enzyme-substrate complex, which is at the heart of a chemical reaction. Oxidation-reduction reactions play an important role in metabolism, or the processes that maintain the balance between building up and breaking down molecules throughout the human body. Oxidation-reduction reactions are coupled reactions, and they're an important part of metabolism, or the process that maintains the building up and breaking down of various materials throughout the body. Oxidizers reduce and reducers oxidize, and oxygen is the great oxidizer, hence the name. Oxygen likes to gain or hoard electrons, otherwise reduce other atoms or molecules. There are essentially 3 metabolic pathways to making ATP. You have 2 anaerobic, or without oxygen, and you got 1 aerobic, or with oxygen. These 3 phases are the ATP-PC, or phosphagen system, glycolysis, or the breakdown of sugars, and then there's the aerobic pathway to metabolism. In a general sense, these pathways occur in succession as intensity or duration of activity increases. ATP-PC is first, glycolysis is second, and finally aerobic energy production. Phosphocreatine levels fall precipitously during the first 3 minutes, with ATP production from PC highest during that first minute. Glycolysis starts contributing during that first minute. As steady-state oxygen uptake is reached, aerobic metabolism provides ATP. So let's start from the beginning. The enzyme creatine kinase catalyzes the breakdown of phosphocreatine, donating a phosphate group to ADP to form ATP. Muscles store very little ATP in phosphocreatine, and in the phosphagen system, ATP production happens only during recovery. This recovery can take less than 5 minutes or more than 15 minutes following exhaustive exercise. The ATP-PC system provides energy for short-duration, high-intensity activities lasting less than 5 seconds. A 50-yard dash, a high vertical jump, a tackle, a quick sprint downfield. As we've touched on, the system is only good for 5 to 10 second bursts of activity, at which point reliance for ATP production shifts over to the process called glycolysis. Glycolysis is the process that breaks down glucose or glycogen to form 2 or 3 molecules of ATP plus pyruvate or lactate. We'll be touching on that in a little bit. With all metabolic pathways, the end goal is something called phosphorylation, which is combining a phosphate group to another molecule, in this case, adenosine diphosphate. The manner by which glycolysis transfers bond energy from glucose to rejoin inorganic phosphate groups to adenosine diphosphate happens in 2 phases: an energy investment phase that costs ATP to start, and an energy generation phase where ATP comes out at the end. The way that this happens is both phases have 5 reactions. The energy investment phase, also known as ATP priming, uses ATP at 2 points to add phosphate groups. If glycogen is used as a substrate, only 1 ATP is needed because glycogen is already phosphorylated by inorganic phosphate. The second phase, the energy generation phase, is where ATP is formed. Phosphate groups are added to 2 adenosine diphosphates at 2 different points, totaling 4 molecules of ATP. Nicotinamide adenine dinucleotide, or NAD+, is a carrier molecule that shuttles hydrogen out of this reaction. NAD+ is reduced to NADH. If oxygen is present, then NADH shuttles hydrogen into the mitochondria, otherwise known as the powerhouse of the cell. If oxygen is not present, then pyruvate, which is formed at the end of glycolysis, accepts that hydrogen and transforms into lactate. Carbohydrate is the only nutrient whose stored energy can be used to make ATP via glycolysis. If glucose and glycogen are not available, then the body can synthesize that through a process called gluconeogenesis. We noted the disparate end products of glycolysis as being either pyruvate or lactate, distinct from one another by the presence or lack thereof of oxygen to accept hydrogen. Scholars of old referred to glycolysis involving oxidation as aerobic or slow glycolysis, in contrast to anaerobic or fast glycolysis, which ends with pyruvate converting to lactate. This misconception was based on the assumption that oxidation could only occur in aerobic processes. Today we know that to be untrue. In fact, if exercise is long or intense enough, lactate is going to be produced. According to Baker, Clare McCormick, and Robergs, the entire glycolytic pathway is oxygen independent or anaerobic. More biochemically correct alternate names would be lactic glycolysis versus alactic glycolysis for intense and steady state exercise conditions, respectively. And now we leave the anaerobic pathways and move into the aerobic pathway, otherwise known as oxidative phosphorylation, involving two cooperating processes, the Krebs cycle and the electron transport. A critical thing to remember going forward is that fat burns in the flame of carbohydrates. We'll be touching on this several times. Fuel for this in the form of fatty acids or glucose can come from within the muscle or from outside the muscle via ingestion or processes in the liver. Aerobic metabolism occurs in the mitochondria, the powerhouse of the cell. Hydrogen is the real star of the show, a lone proton with a single electron. We noted that the two cooperating processes of aerobic metabolism are the Krebs cycle and electron transport chain. Acetyl-CoA generated from substrate degradation is oxidized in the Krebs cycle for use of forming ATP at the end of the electron transport chain. The purpose of the Krebs cycle is to remove hydrogen and its associated energy from substrates. This occurs by breaking pyruvate down into acetyl-CoA, a process that also releases carbon dioxide. Acetyl-CoA is then combined with oxaloacetate to form citrate. This cycle of regenerating oxaloacetate to form citrate and carbon dioxide results in the formation of NADH and FADH, which carry electrons into the electron transport chain to synthesize ATP. Put more simply, acetyl-CoA enters the Krebs cycle, and through various processes, as it's broken down and converted into citrate, what is also released are two carrier molecules, NADH and FADH. These go into the electron transport chain. On the other end of this, oxygen accepts a lone electron, and hydrogen turns into water and moves on. ATP is formed from this entire process. And though it's part of an aerobic pathway, the Krebs cycle does not actually use oxygen, although oxygen is used as a hydrogen acceptor at the very end of the electron transport chain. We mentioned that the Krebs cycle oxidizes acetyl-CoA after it has been produced by the breakdown of substrates. This key 2-carbon molecule can be made from carbohydrates, fats, or proteins. After pyruvate is formed via glycolysis, it can be broken down into carbon and acetyl-CoA, which can then enter the Krebs cycle. Amino acids, the building blocks of protein, can also be converted into pyruvate, either through direct conversion or through a process of breaking down protein called proteolysis. Approximately half of the free amino acid pool already exists within the muscle tissue, and that proportion can increase as exercise intensity increases. Several amino acids can be converted through various processes into pyruvate, including alanine, which can be directly converted. Through lipolysis, fat can be broken down into free fatty acids. After following a process known as beta oxidation, these can be converted into acetyl-CoA. The mechanisms involved are different based on the substrate in question, but the end result is always the same: the formation of acetyl-CoA to be used in aerobic metabolism, otherwise known as oxidative phosphorylation. Phosphorylation is simply the process of adding a phosphate group to another molecule. In this case, that molecule is ADP, adenosine diphosphate. That's the end goal of all metabolic pathways, to reunite ADP with inorganic phosphate to create ATP. In the ATP-CP system, this is a fairly straightforward process with a single enzyme, creatine kinase. Glycolysis is a substantially longer process, but the end result is 3 to 5 times more ATP than what is made by the phosphagen system. Oxidative phosphorylation is a fairly drawn-out process, but as long as acetyl-CoA and oxygen are readily available, the process will continue dishing out 32 to 33 molecules of ATP with every turn of the wheel. The Krebs cycle's purpose is to remove hydrogen and its associated energy from substrates. In a nutshell, for every 2 glucose in glycolysis, 2 pyruvate are formed. In the presence of oxygen, 2 pyruvate are converted to 2 acetyl-CoA. Each glucose molecule results in 2 turns of the Krebs cycle. During each turn of the Krebs cycle, 3 NADH and 1 FADH are formed, the equivalent of 2.5 and 1.5 ATP respectively. Additionally, 2 ATP equivalent guanosine triphosphate or GTP molecules are formed. This is substrate level phosphorylation. This accounts for only a small amount of energy conversion because most energy yield from NADH and FADH is taken to the electron transport chain to form ATP. And that takes us to the electron transport chain, which uses the potential energy of reduced hydrogen carrier molecules to rephosphorylate ADP into ATP. Glycolysis degrades glucose, yielding 2 NADH per glucose molecule. Pairs of electrons from NADH and FADH formed by the Krebs cycle pass down the electron transport chain by cytochromes via a series of oxidation-reduction reactions at 3 places, otherwise known as pumps. FADH bypasses one pump, yielding 1.5 ATP, while NADH yields 2.5. At the end, oxygen accepts electrons and combines with hydrogen to make water. Without oxygen, oxidative phosphorylation cannot occur, and ATP can only be formed by anaerobic metabolism. The chemiosmotic hypothesis is the mechanism that explains aerobic ATP production. As electrons are transferred along the cytochrome chain, the energy released is used to pump hydrogens released from NADH and FADH from inside the mitochondria across the inner mitochondrial membrane, resulting in the accumulation of hydrogen throughout the intermembrane space. The potential energy of this accumulation is used to recombine phosphate with ADP to form ATP. The enzyme ATP synthase catalyzes this phosphorylation of ADP via the accumulated hydrogen. If the last cytochrome, cytochrome A3, remains reduced from the added electron, it would be unable to accept more electrons, and the electron transport chain would stop. Oxygen takes that electron and keeps the process running. Oxygen combines with 2 hydrogens to make water. As stated earlier, the ATP-PC system generates 1 ATP for every turn of the process. Glycolysis, on the other hand, will generate 2 to 3 ATP depending on whether glucose or glycogen are used in the process respectively. In addition, as NADH shuttles protons into the mitochondria, an additional 5 ATP are formed. Not bad numbers, right? However, when aerobic pathways are involved, that shoots up to 32 to 33 molecules of ATP. When pyruvate from glycolysis is converted to acetyl-CoA, 5 additional ATP are produced. 2 ATP equivalent guanosine triphosphate molecules are formed with 6 NADH and 2 FADH molecules, a combination of carriers that enters the electron transport chain to yield an additional 18 ATP molecules. That brings our grand total up to 32 ATP. 33 of glycogen was used in glycolysis. Oxygen accepts electrons and combines with hydrogen to form water, and the process keeps on cycling. Again, it all starts in the anaerobic pathways. Without them, aerobic phosphorylation can't even kick off. Remember, fat burns in the flame of carbohydrates. Lactate threshold is the point at which there is a sudden increase in blood lactate levels, indicating that lactate production is surpassing the removal of lactate. The factors that influence the lactate threshold are low muscle oxygen, accelerated glycolysis due to epinephrine and norepinephrine, recruitment of fast-twitch muscle fibers, and reduced rate of lactate removal. Low oxygen levels seems like a likely culprit for high lactate levels, because without oxygen, pyruvate has to convert into lactate. Glycolysis can also be accelerated due to increases in epinephrine and norepinephrine. These hormones increase glycolytic rate, and NADH production exceeds capacity of shuttle mechanisms that move hydrogen from sarcoplasm to the mitochondria. In this scenario, pyruvate accepts hydrogen and converts to lactate. Lactate dehydrogenase, or LDH, is responsible for this conversion. Recruitment of fast-twitch muscle fibers is also a factor, as lactate dehydrogenase has an affinity in type 2 muscle fibers for attaching to pyruvate and forming lactate. Interestingly, in type 1 or slow-twitch muscle fibers, LDH has the exact opposite affinity. The reduced rate of lactate removal is another factor that impacts the lactate threshold. The liver can use lactate to synthesize glucose in a process called gluconeogenesis. Also, there is a system called the lactate shuttle. Type 1 muscles and the heart can oxidize lactate. Remember, LDH in type 1 fibers converts lactate to pyruvate, which in turn gets converted to acetyl-CoA in the Krebs cycle. So carbs or fat, in general, carbohydrates are going to be used as a substrate in activities of high intensity or long duration, whereas fats will typically be your primary substrate for low intensity, long duration exercise. The crossover point is the intensity at which you primarily are burning carbs. This is based off of your VO2 max. An important note is that endurance trained athletes typically tend to use a higher fat to carb ratio than less fit athletes do. As touched on, the crossover point is the intensity at which carbohydrate metabolism exceeds fat metabolism. So what are the reasons for this shift? One is the recruitment of fast twitch fibers. These fibers have an abundance of glycolytic enzymes, but very few mitochondrial or lipolytic enzymes, which are responsible for fat breakdown. Another reason for this is an increase in blood epinephrine levels. This triggers an increased activity in the enzyme phosphorylase, which will then increase glycogen breakdown, thus increasing the glycolytic rate and lactate production. So there's been a lot of talk about carbs, but what about fat? The switch to fat metabolism is regulated by variables that control lipolysis, or fat degradation. Enzymes called lipases break down triglycerides into free fatty acids and glycerol. Lipases are inactive until stimulated by epinephrine, norepinephrine, and glucagon. During long, low-intensity exercise, blood epinephrine levels rise, increasing lipase activity and promoting lipolysis. Lipolysis is slow, and increased fat metabolism only happens after several minutes of exercise. Free fatty acid mobilization is inhibited by insulin and high levels of lactate. Insulin directly inhibits lactate. Normally, blood insulin levels decrease during prolonged exercise. However, high-carb meal or drink 30 to 60 minutes before exercise increases glucose levels and triggers more insulin to be released from the pancreas. So as insulin goes up, lipolysis goes down, which decreases fat metabolism and increases the reliance on carbohydrates. We've said it before and we'll say it again. Fat burns in the flame of carbohydrates. During short-term exercise, muscle stores of glycogen or blood glucose levels would not be depleted. However, during prolonged exercise, muscle and liver glycogen can reach severely low levels. Depletion of glycogen leads to fatigue. Simply put, less carbs means less pyruvate. Less pyruvate means a reduction or even termination of the Krebs cycle, which leads to no ATP, which leads to fatigue. Pyruvate is a product of glycolysis and a precursor to several Krebs cycle intermediates, such as oxaloacetate and malate. Reduced glycolysis also reduces levels of pyruvate in the sarcoplasm and of Krebs cycle intermediates. This slows down aerobic ATP production, limiting performance and leading to fatigue. Fat can only be metabolized via Krebs cycle oxidation, meaning that depleted carbs results in reduced fat metabolism. Protein can also be used as a substrate in metabolism, but first it has to be broken down into amino acids. Amino acids are available in the free amino acid pool located in the muscles and the blood. Skeletal muscle can directly metabolize branch-chain amino acids, and the liver can convert the amino acid alanine directly into glucose. Protease is the enzyme responsible for protein degradation, otherwise known as proteolysis. Increased calcium levels from contracting muscles can activate protease. Substrate sources. Sources of carbohydrates are in the muscle and liver. Blood glucose is primary fuel for low-intensity exercise. Muscle glycogen is primary fuel for high-intensity exercise. Prolonged exercise stimulates glycogenolysis to break down glycogen and release glucose into the blood. Sources of fat in the body include fat cells and muscle cells. Use of fat as a substrate depends on its availability to muscle cells. Triglycerides must be degraded to free fatty acids and glycerol via lipolysis, and then free fatty acid can be converted to acetyl-CoA and enter the Krebs cycle. Between 65 to 85% VO2 max, fat contribution as a substrate is about equal between free fatty acids and triglycerides. Over half of the free amino acid pool is available in muscle cells, and that number simply goes up with exercise. Proteolysis can make more amino acids available by degrading protein, which is essentially what all tissues in the body are made up of. Fortunately, protein typically only plays a small role near the end of extended activities. and its contribution does not have any detrimental health effects. Last but not least is lactate. You remember lactate, the end product of glycolysis when oxygen is not available, forcing pyruvate to change its ways. Well, contrary to old ways of thinking, lactate is not the enemy, and it turns out it is actually a friend. In fact, lactate is an oxidizable substrate during prolonged activity. Type 1 muscle fibers in the heart can oxidize lactate. Remember, lactate dehydrogenase, LDH, in type 1 fibers converts lactate to pyruvate, which in turn gets converted to acetyl-CoA in the Krebs cycle. The liver can use lactate to synthesize glucose via gluconeogenesis via something called the Cori cycle. Without the Cori cycle to take up blood lactate to form glucose, hypoglycemia would occur during prolonged exercise. So that's energy metabolism. It's a lot of information to take in, but here are the key points. Adenosine triphosphate is the king, and breaking its phosphate bond creates energy for muscular contraction. At the end of the day, the purpose of all metabolic pathways is to reunite adenosine diphosphate with inorganic phosphate to combine them to form ATP. Think of the pathways as being in a queue of sorts. The ATP PC system comes in very early and does its job, but that job cannot go on for very long. Glycolysis takes over, and for short or high intensity activity, it might be all that you need. Emphasis on might. Finally, there's the big one, aerobic phosphorylation. Here, the Krebs cycle and electron transport chain combine forces to take in acetyl-CoA and make some more of that sweet ATP. Plus water. Sweaty, sweaty water. We've said it so many times already that it just wouldn't feel right to not repeat it one more time. Fat burns in the flame of carbohydrates. So there it is. Metabolism. It's all about making ATP, breaking ATP for energy, and then reuniting adenosine diphosphate with a phosphate so that adenosine triphosphate can be put back together just to break it again for energy. And that's what it all comes down to. There's a lot of terms, there's a lot of information, there's a lot of essential concepts to understanding what phosphorylation is, how it works. So just keep it easy. Eat, work, play. What else is there? There are several methods available to use substrates other than carbohydrates to drive aerobic metabolism, but those methods all have the same purpose, to replace carbohydrates with, well, carbohydrates through a process called gluconeogenesis. Lipolysis and beta oxidation both have a role to play in burning fat to drive aerobic metabolism, but that process is slow to start and slow to manage. Glucose is the kicker that gets the whole party started. Sports drinks have been shown as an easy means of replenishing glucose while exercising or playing sports. And it is true that there is evidence that supports their effectiveness at improving performance. But there's also evidence that carbohydrate feeding at the onset or during activity can have a negative impact on glucose production in the liver. In 1999, Juke and Drop and Company studied the effects of ingesting liquid carbohydrates of a 4% and 22% glucose concentration. And as we hinted at, the low and high concentration drinks actually reduced or even fully suppressed the production of glucose, respectively. Asker Jukendrup later joined forces with Sophie Killer, real name, to ultimately determine that conclusion that he and his team reached in their 1999 study was unfounded. But this just goes to show that science is constantly developing, and we continue to learn as well as unlearn, and even the information shared in today's video could be deemed incorrect tomorrow.